All right, now it's time for part 5 of structural immunology. I'm going to try to do this one fast because first of all, I have about half an hour that I want to say to get you biochemists ready for our virtual tour on Friday. I've already talked long enough about all these other things, and but I need to talk about one of the other branches of the immune system. The other thing is, in 30 minutes, we're supposed to start cooking dinner, and I need to fire up the grill. So I'm going to try to do this as quickly as I can, knowing that you can rewind, okay? So here we go. We're going to talk about T cells and MHC interactions. Antibodies were molecules that can adapt and detect new antigens, new invaders in the body that are in the blood outside of cells. Okay, but what about the invaders like a virus that get into the cell? Well, you actually have a system that looks inside a cell and can see both viruses and turn cells into tumors inside the cell. This is a whole nother system. It interacts with the B cells, but it's called the T cells. And it involves a window into the infected cell. Remember that B cells and T cells were cousins when we looked at them, how they developed way back in part 1. In fact, that still shows up because T cell receptors are cousins to antibodies. For example, they're all made of these Ig domains. So remember how we had all those beta sandwich domains in antibodies. We have the same exact domains in TCRs, T cell receptors. But they're even arranged somewhat similarly. They look like one branch of the antibody. Remember, the antibody had the antigen binding site at its tip. It had 3 loops in each, times 2 equals 6 loops total. T-cell receptor has the exact same architecture for its binding site. So everything that we saw about how antibodies bound antigen, especially how antibodies bound protein antigen, still applies to what we're doing with the T-cells. When a virus infects a cell, we've seen pictures like this before. These are pictures of viruses actually infecting a cell, taking over them. You can see them sort of popping out of the cell if you look on the southwest over there. But what T cells can do is that they actually bind to the surface of a cell, and somehow they know that inside the cell there is a virus. How do they know that? They're binding something on the surface that reflects what is going on inside the cell. And you see down here, actually, to my left here, we have a little T for the T cell and a V for the antigen virus infected cell. There's an interface between them. At that interface, the T cell is recognizing the virus shape somehow. How it does that is a fascinating piece of biochemistry, and that's what I want to tell you about. Realize that virus means something is wrong inside the cell and replicating out of control. Well, what if the cell itself is replicating out of control? There is also something wrong inside the cell in that case. And in fact, the T cell can detect that there's something wrong inside the cell for a lot of the different viruses and for cancer cells as well. So there is actually cancer immunity. There's cancer immunotherapy where you can actually put T cells into a person that are reactive against cancer. You can try to make the cancer cells more visible to T cells. And you can even do what's called a cancer vaccine, where you put one of these proteins that says to the rest of the immune system, says to the T cells, hey, something's really wrong in here because there's a lot of this going on because there's a lot of cancer going on inside me. That's what a cancer vaccine is. So if you think that vaccines are only for infections, there's actually a possibility that they can be for cancer as well. Vaccines are about educating the immune system. And so you can educate the immune system against a particular pathogen or against a particular kind of tumor, if you can find a signal that works for that. So how do T cells see inside other cells? Well, there's actually a little conveyor belt. And if you imagine, I don't know if this is like in a forgery or like, the things where they're moving buckets of liquid from one place to another. I'm just remembering something where you kind of have a wheel of buckets, and the buckets are constantly going down and dipping into the water and like pulling it back up. You have that kind of thing. It's like a water wheel, I guess. Where you have the water being moved up by different buckets. That's the same exact idea, except you have buckets inside the cell that are constantly moving up to the surface that are carrying along proteins from inside the cell. What's really cool is that this is a system that's called the HLA or MHC system, and T cells detect what is in the buckets, okay? So that's how T cells can see inside other cells. There's this mechanism When a virus infects a cell, one of the things that it does is it takes over the cell, makes a lot more virus. There's a lot of viral proteins now inside the cell. Well, now we have these buckets. MHC Class I is what's shown as the buckets right here. They're in yellow. And they bind the red viral proteins. And then they get moved to the surface of the cell, and they're a bucket holding the viral protein on the surface of the cell. The T cell receptor can then come along and say, hey, I've never seen that little red thing before. Time to call the rest of the T cell brigade, okay? So you have that thing going on constantly, and it's the MHC system, also called the HLA system. The other thing to notice is that this is just one of those kinds of systems. We actually have two systems of buckets that are offering windows into the cell. The thing that you'll see that these are peptides. Peptide is a name for a short fragment of a protein. Because proteins are so big, you need it to fit into the MHC bucket. And so there's actually systems in the cell that just chop up everything in certain areas. And they chop up everything. If there's a bunch of viral proteins there, you get a bunch of viral peptides. And the peptides are small enough to fit into the MHC bucket, and then small enough to be displayed on the cell surface. It's a really cool system. It's pretty amazing. So the T cell receptor will surveil the MHC buckets on cells. And it's used to your normal MHC buckets and your normal retinue of peptides that's presented on those buckets. If everything's normal, the TCR has been educated to not freak out, hopefully. If you have a T cell that's freaking out in the case of a normal situation, you have autoimmune disease. So the thing is, if the TCR sees something in a bucket that is never seen before, if it sees a peptide that is not seen before, it says, uh-oh, something's wrong. Something is not normal. Therefore, I'm going to bring out all of my weapons against this infected cell or this transformed cell, and I'm going to destroy it. In this way, the T cells are cytotoxic in this sense because they're cytotoxic against targets that are expressing peptides that the TCR has never seen before. So the TCR has to recognize the bucket, it has to bind to it, and it also has to recognize the normal shapes of normal peptides so that it can recognize when there's an abnormal shape. Okay? How that all works, amazing system. We don't have time to talk about it all right now. But remember, we're talking about this branch of the immune system that basically is armed. They're like the police force of the immune system. They're patrolling and they're knocking on doors to see if the virus is inside there. And so that's where the cytotoxicity comes in if they detect that there's a virus, if they look through the window and they see inside the cell. And so this is the weapons that the cytotoxic T cell has. You see I have the T cell in blue, and this is one of my, remember my favorite artist, David Goodsell, for protein paintings. You have the cytotoxic T cell in blue on the top, and you have the target cell expressing MHC in orange on the bottom. And if you look way over there, you see like a blue T cell receptor that's binding a little red MHC molecule. By the way, you also see a little green receptor that's binding the side of the red receptor on that side of it. It's going to be to the right of the blue TCR, if you can find the blue, red, and green that's coming together. That green co-receptor is the CD8 co-receptor. CD8 is one of the signs that this T cell is a cytotoxic T cell. And when that engages, you see that engages, the T cell is activated, and the T cell has these little bubbles filled of nasty things. And it brings those bubbles to the surface, and it basically spills those out onto the target cell. It has something called perforin that actually punches holes in the cell. You can see the pink holes in the cell, in the orange cell being punched right over there to my left. And you see the perforin, it has granzymes, which are enzymes that eat up the cell. And it also makes a bunch of reactive oxygen species. Remember how oxygen has a connection to immunity, both to give it power and to serve as a reactive weapon. They deliberately make this nasty form of oxygen to throw at the target cells. And I think that's pretty key, and that's pretty interesting as well. So you have these systems. They will defend against all sorts of infections. You can have a bacterium that infects us, or actually there are certain cells. This is another system that uses the buckets, and it looks similar, but notice that there's some differences here. Here you have a bacterium that's outside the cell. The cell reaches up and it grabs that bacterium, and then it puts it onto a little yellow bucket. It cuts it up, puts it on a bucket, and puts the bucket on the surface. But this bucket is an MHC Class II bucket, and it is for a system, for a special cell that is the macrophage. Remember, we saw those before. They're the ones that are able to eat and engulf other cells. They patrol around looking for bacteria. So they have actually adapted to be able to use MHC Class II to activate a different branch of the immune system against external bacteria. There's also are involved, Class II MHC are also involved in antigen responses. On a B cell, it has Class II MHC because when a B cell the B cell surface receptor is exactly like the antibodies that it makes. When it binds antigen, it takes that antigen inside, chops it up, puts it on the MHC Class II. In both of these cases, the MHC Class II is binding a pathogenic peptide, but it doesn't want to be killed. You know, it's not cytotoxic. It's not actually infected. It has deliberately brought this in, put it in the bucket, and so it puts it in a different kind of bucket that activates a different branch of the immune system. Okay? So MHC Class II molecules are actually a different system, works the same at a molecular level, but it's separate at a functional level. And they're associated with not cytotoxic T cells, but T helper cells, because they don't kill other cells, they activate, they check the stuff on the surface, and they say, hey, macrophage, you've got a bit of bacteria protein in your teeth, you know, in your MHC molecule. So then they send that a signal saying, that's weird, you should be activated and you should go against bacteria in the area. For the B cells, they say, hey, you should be activated and you should make a lot more antibodies. So you see that these branches of the immune system are actually intertwined. Cytotoxic T cells are for all cells that have MHC Class I, looking for viruses and tumors. The specialized cells, the immune system cells that are trying to have specialized functions, they are MHC Class II. A subtle difference between the two, and different shaped molecules that work slightly differently. And so these are like radios. T helper cells are like the radios that they don't kill, they aren't armed, but they send messages that say, activate the immune system, and hey everybody, come over here, and we're going to all fight this thing. By the way, these messages sometimes are associated with cytokine storms and things like that. So all these branches of immunity are interrelated in cycles of cells activating and deactivating, and B cells have to check in with T helpers to be able to activate. So we're going to talk mostly about the Class I MHCs, but we have a few little stories about Class II. Structurally, they're very similar. T cell receptors all look like this. They're all two chains, alpha chain, beta chain, two Ig domains each, beta sandwich Ig domains, and then they all have a little bit of glycosylation, carbohydrate on them, and they have one end that binds antigen, it's a variable region, and the other end that has a constant region, and then that's basically it. They're kind of like one arm of an antibody. And so the business end of the TCR, if you look at it, here's an example. You can see the Ig domains. You can look over here and you can see, if you look all the way above my head right here, you can see that there's 6 colors there, basically. There's 6 loops, 3 for the heavy chain, H, 3 for the light chain, L, and by the way, they still call it heavy and light, even though there's complicated reasons. Anyways, but they have 6 loops, basically, 6 colors, 6 loops, just like antibodies. And these loops engage the MHC peptide complex. So this is what they engage. They engage the target cell, and this is a peptide in red shown in an MHC, and you can see that the MHC looks like it has this little peptide binding groove. In fact, that's its whole job, to be a bucket for peptides. And so I've described this as a window, and I've also described it, and I did this when I was describing my thesis. In my thesis defense, I described the MHC as a hot dog bun that holds the hot dog. I think that goes with the fact that this is like the right color for a hot dog right here. But it's really just a bun, but it also has its own surface. The MHC surface will engage some of the TCR loops, and the peptide will have a few side chains sticking up that also will engage the TCR. So the TCR has to feel this surface, and it has to detect, okay, number one, Here's an MHC just like what I expect. Oh, wait a second, what's this peptide in the middle? I haven't seen this before. That's what a TCR has to do. And it's important to remember that we have multiple molecules coming together here. This is rather complex, but these are just molecules and they are just amino acids. So again, we're in my home court here. I'm a protein chemist. I work with amino acids and proteins, and I do chemistry with them. I can do chemistry with this, and I did do chemistry with this. It's how I got my PhD. So a side view of the two different types of MHC proteins. Remember that we have class 1 and class 2, and actually these are sort of reversed. Unfortunately, class 1 is on the right, class 2 is on the left. You can't have everything. But when you look at it from the side, you can see that they have two Ig domains underneath, the beta sandwiches that you see all over the place. And on top they have an interesting sort of thing that has two alpha helices between which the peptide sits. That's the peptide binding site. That is the hot dog bun. And so for both MHC class 2 and MHC class 1, from this view, they look really, really similar. Let's take a closer look at both of the classes. MHC class 1, here's three different views of it. And you can see that it has one chain that goes, there's alpha 1, alpha 2, alpha 3, light blue, green, yellow. That's all one chain, which includes the peptide binding cleft, and then one of the Ig domains below. What's interesting is MHC class 1, its fourth domain, is separate chain. And so it has to bind there. So technically MHC class 1 is really two chains, two molecules. Beta 2 microglobulin is separate from the other three. But all that is to say, you can see the peptide binding cleft is up there. If we can make this molecule, we can use it to activate the immune system. So when we look at the MHC class 1 peptides, here's a bunch of peptides that bind the same MHC. And if you look, you see that they, they're a little bit messy, but they overlap, really. And especially if you look at the two places that point down. There's a leucine pointing down and a valine pointing down in all three peptides. They have different residues pointing up and different residues pointing side to side. And by the way, also you'll notice they're the same length. They're each 9 to 10 letters long. And this makes sense because remember the MHC bucket, the leucine and the valine are pointing down into the bucket. And so the bucket has two little pockets that bind leucine and bind valine. These don't have to fit perfectly, they just have to fit well enough to work. So if you take a bunch of MHC class 1 peptides, you can eventually see that they all have a pattern. All the peptides that bind a particular MHC class 1 should have similar anchor residues, which are fitting into the MHC pockets. And you see right here we have, these are all 9 to 10 letters long, and the anchor residues are shown in green. And if you're a biochemist, you know that these letters are similar. L is similar to V, Y is similar to F. If you don't know that, take my biochemistry course. I offer it in the fall. But you can see that they have these patterns. Now the patterns for the non-anchor residues can be very different. You have a lot of different letters there. But the letters pointing down have to be the same. The other thing about these, they're all the same length, or very similar, 9 to 10 letters long. So when you look at them in the context of the peptide binding groove, you can see that not only do you have the two pockets that bind the two residues, you also have a bunch of hydrogen bonds that form special cyan bonds that tie it down. And then you notice that the hydrogen bonds here are focused at the ends of the peptide. At the left end and the right end, you see these little cyan bonds. Because all the peptides are going to be chopped off, they're all going to have hydrogen bonding motifs or moieties right there. And so they'll just use that as ties to tie down any kind of peptide that comes along. The important thing about these, all peptides should be able to hydrogen bond about the same. You don't want picky buckets. You want buckets that will bind a lot of different things. And so these are buckets that will bind a lot of different peptides, because that's what you want. MHC class 2 looks a lot like MHC class 1. You've got two domains and two chains. The only difference here is that you've got two equal chains, that you have the peptide binding cleft is split among the two chains. But other than that, from this perspective, it looks pretty much the same. You might notice there's something a little different about the peptide binding cleft. It might look a little more open at both ends, because that's the main difference between MHC class 2 and MHC class 1. When you look at the peptides, here's MHC class 1 on the left, MHC class 2 on the right. Well, MHC Class II is like the bun-sized hot dog bun. MHC Class I is like the little Oscar Mayer tiny little hot dog that's lost in the bun. You know, it fits entirely inside the bun, and it's even closed at both ends. MHC Class II is hanging out the bun. It's like the one when you're going to the Mariners game, and you get the big old hot dog that they have on the way in. I miss those. I hope that we can have Mariners games sometime in the future. I just don't want to win too much. But I predict that will happen sometime, okay? MHC Class II is open at both ends. The peptides, therefore, are longer. And you can see this if you take a bunch of different MHC Class II molecules. You can see that they bind these peptides, and the peptides can be varying lengths now because they can dangle out the end. The top, by the way, is the peptides that bind a mouse Class II. The bottom are the peptides that bind a human Class II. But Class II also has the pocket residues, the anchor residues that are shown in the colored circles. Again, you see that one anchor, the anchor for the residue numbered 4, is all biochemists will recognize negatively charged residues. The pocket in anchor 9 appears to be a little less picky. And so you want a bunch of different peptides to bind, and MHC Class II will bind longer peptides. But the same idea. And so MHC Class II will also form hydrogen bonds, but it can't really do the same thing as Class I, where it binds the ends, because there aren't ends of the peptide to bind. They're dangling off like this, right? So instead of having hydrogen bonds to each end, they have a bunch of hydrogen bonds distributed across the peptide. And you see you have hydrogen bonds at the N terminus, hydrogen bonds in the middle, hydrogen bonds at the C terminus. And you see that they're distributed. And again, all peptides should have these hydrogen bonds, and if you look at the crystal structure, all the hydrogen bonds look the same. But do they act the same? We don't know until we test them. And that's what I did for my graduate studies. I tested the hydrogen bonds in a mouse Class II molecule. I made the molecules, and I tested them with physical chemistry, actually. I tested the kinetics of binding. And here's my thesis right here. If you want to talk to me about it, I can do another 5 hours of lectures on it, I'm sure. You can look it up. I mutated mouse Class II MHC, and I removed hydrogen bonds systematically along the length of the molecule, and especially at one end versus the other, the N-terminal end versus the C-terminal. And I found out that the N-terminal ones had a much bigger impact. It didn't care that much if we mutated the C-terminal ones. If you mutated one that looked exactly the same at the N-terminus, had this big effect, like it really mattered. And so we said that the N-terminus looks more important than the C-terminus. I also did some mutations in the pocket, and we found out that those actually didn't communicate very much with the hydrogen bonds. It's mostly about the hydrogen bonds, which again, you want a bucket that holds a lot of different peptides. All peptides have hydrogen bonds. Not all peptides have the same anchor residues. And so there is an interplay going on here. And actually, what I really like, I did all this with the molecules. And then other people went in and they looked at how the immune system actually works. And they found out that part of the immune system that helps the peptides load into Class II, it only works on one end of the protein. It works on the N-terminal end, which is exactly where my work predicted that they would have gone. The N-terminal end is the one that is more important. So you have this thing, and you have this T cell receptor PMHC, which is going to go. So I'm going to go through a few examples real fast of how we can use this TCR peptide MHC interaction. This is an interface made of loops interacting with a peptide MHC complex. These are all atoms. It's a complex, flexible group of atoms, but we can understand these from atomic directions using our biochemistry. And so that was a Class I interaction. Here's a Class II interaction. And this is really fuzzy. These are really old because they're kind of the original crystal structures. But the atomic interactions will determine if the TCR engages and if it recognizes that peptide as being something novel, which is the case in which it activates the immune system. So here's the papers I want to talk about. So if we know the shape of an MHC and we know what kind of peptides go in, we can predict which peptides will fit into that MHC. If we can do that, we might be able to predict, hey, if these peptides fit into the MHC, these might be the parts of the protein that the immune system will react to, because that is what determines whether a T cell can react to it. And this is a protein that did exactly that for the SARS spike protein. Now you'll notice I don't have the date on here, but this is from about 10 years ago. So this is definitely SARS-1, right? SARS-2 did not exist then. But SARS-1 does have many similarities, and this should work for SARS-2. This is called molecular mimicry, because remember, the peptides are only so long, and they're only putting a few side chains up against the TCR. And so that means that you can have similar peptides, and they can be different at all the other places, but they can be similar at just the parts that stick up. And they found that there are peptides in the SARS-CoV-1 spike protein that look like they have the same shape presented to the TCR as certain human proteins. In fact, the human proteins they look like are the receptors that they engage the ACE2 receptor. So this makes perfect biochemical sense. No, I just want you to notice right here, they used an algorithm. This algorithm isn't necessarily very smart, it's sort of statistical. They take all the peptides that they know binds a certain MHC, and they basically just take the statistical patterns of those peptides. And they say, does this sequence match those patterns or not? If it matches those patterns well, then I'll say I think it binds the MHC. It's not necessarily terribly accurate. Reported accuracy of this method is about 75%. But it's a simple way to narrow down the field. So what they did is they took the spike protein, as shown here, and they found the areas that are in yellow are those that are predicted to bind MHC. The areas that are blue are the ones that share sequence with the human proteins that are known to engage the ACE2 receptor. And the purple are both. I would have made it green, honestly, because yellow plus blue makes green, right? But there's no accounting for taste. There we go. We have the blue ones showed up in the computer scan as binding MHC, and they also showed up in a homology scan as sharing sequence with human proteins. Let me show you exactly right here. This little stretch of peptide is purple. So the computer predicted it would bind MHC, and they also found out that this purple stretch matched this exact part of it was the S protein that they tested in the computer, but it looks very similar to this, oh boy, I forget the name, angergum, I can't, they don't have the full name there. But whatever this protein is, it is a natural ligand for ACE2. So it implies that this is a part of it that engages the human protein, that engages the ACE2 receptor. It's possibly also a part of this S protein, the spike protein, that engages the ACE receptor. And they found this for two different proteins. But this particular peptide, they actually made these peptides and they tested them against T cells. And here's the amino acid sequences. And here's D08, and you can look it up and you can see that that sequence is in both the spike protein and in the natural binder of ACE2. And so right here is where they showed it, and they reacted it with antibody sera from SARS-1 patients. And this sequence is found both in the spike protein and in the ACE2 ligand. By the way, I might have misspoken earlier and said it activated T cells. I forgot, this one is actually activating antibodies. So this is even simpler than that. This is just finding a shape that mimics a natural antibody. And we have an evolutionary reason for that. The receptor is what they both bind. And so they've evolved to bind the receptor. The good news is antibodies against the protein will also bind antibodies, will also bind that human natural ligand. And actually, I said that's good news. It's complicated, right? Because it could be the source of an autoimmune response later. All this matters here is if the sequence is similar, then the immune system can react to both a human protein and a viral protein. It doesn't know that one's from virus. It just knows they have the same shape. And you can see the same shape there. This leads to some really interesting effects, and this is one of the things that I really want you most to know. There are some T cells that they raised against HIV. They react to HIV peptides, and these are T cells, so they're reacting to peptide MHC complexes. And then they found something funny. They ran those same T cell clones. They have clones 1 through 5 that are all the way over there. And wherever you see a plus, that clone also reacted against the other peptide. Look at the names of those peptides. A. tropicalis, P. marinus, L. bacterium. These are not HIV peptides. And the sequences, if you look into the sequences, you can see some similarities structurally that these little peptides happen to have chemical similarities because, again, there's only so many amino acids and only so many spaces in a peptide. They have similar shapes to what the HIV is used to reacting to. These recognize HIV peptides. They also recognize these diverse new peptides. This is not because they've ever seen them before. This is just because the shapes of the HIV happen to match the shapes of these tropical diseases. So somebody with immunity to HIV that has one of these T cells would also have some immunity to the tropical disease, and vice versa. It's really complex, but I just want to show you that it's possible. There are memory T cells that you have, and so I wonder if something like this is going on at some small extent with SARS-CoV-2. Obviously, there's a large proportion of the population that gets infected, but I wonder if there are some sectors, even though they've never seen this virus before, that they have some cross-reactive immunity from something else that happens to have the same shape. We just don't know until we find it. And so there are some good surprises out there. This also goes with the idea that children don't have as many different T cells yet. As you develop T cells against one thing, you actually end up with T cells that react against another infection. And this might be why kids are always sick. Their immune system is still learning. And one of the ways an immune system can learn is a vaccine, where you deliberately give it just the peptide or just a broken part of the infection. And that will provoke T cells because all it needs is the peptide. The peptide by itself has a shape that the T cell needs to learn. And so this is one where flu vaccination was actually producing T cells that reacted against it. Here's a hemagglutinin H1N1 sequence, and you see that it also reacted against these two other things on the bottom. Those are very different organisms, but you can even look up and down and you can see they have the same sequence in the middle. Peptides are only very limited in what they can be, and there's going to happen to be some peptides from infection 1 that are also shared by some other infection out there. Here they found some examples. How much does this go on? I don't know. But I'm saying that it goes on. And it would be something very interesting to look for as we go along. What kind of people have immunity against this? So the one last story I want to tell you, this just came out on Friday. And we're actually going to talk about this story when we get to our tour. Remember that the MHC has 3 parts. The heavy chain for class 1, beta-2 microglobulin, and the peptide that binds on top. So if we want to make that, we can make all 3 pieces and then mix them together, but they won't come together very well. What's better is to make them all as one chain and to let them fold together and sort of tie them together, like, "Oh brother, where art thou?" when they're sort of on the chain gang and they're tied together. They can't get away from each other. It'll work better as a reagent. And this is called a PMHC, an SCT, single chain trimer of the MHC. This is a side view of one of the early trimers that was developed. This is a review from the person that developed them, one of the labs that was primary in that. And you can see you had the peptide in yellow way on the end. You had a linker, beta-2 microglobulin, linker, and then the heavy chain. And you can make that as a single construct that will then carry on. These were used by the virtual tour that we are going to take. We're going to see the lab where these were used and where part of this paper was actually done. This was published in March 8, 2020, and right there is Mary, who is going to be our tour guide on Friday. So I'm going to talk about what she did. So the abstract has 3 parts, and I want to just show you these 3 parts. Big complex paper, but there's a simple uptake from it. They basically didn't use a computer. Well, they used a computer to start with, but they actually used lab work to find what are the antigens that are presented on PMHC for SARS-CoV-2. And so they actually found these, and they showed that these activated T cells, and they even found that they activated T cells in SARS-CoV-2 patients. So what they did is they made a library of all the possible peptides that they thought had a good chance of working for these single-chain trimers. And then they did the other things. I'll go through the other ones. They detected the evolution of virus-specific T cell populations. Basically, they detected virus-specific T cells in patient populations by using this, by finding out which peptides are actually used by the immune system, which peptides go in the buckets. Okay? So they made a bunch of buckets. This is a really complicated thing, but this is how they made a library of buckets. First, they took the sequence of the spike protein. They used a computer program to find out I forget how many, it's like 100 or so of the best sequences. And they made 100 or so plasmids that had all of those peptides in them in MHC molecule as a single chain. They went through, there's the library, and you can see the different peptides stuck into the MHC. They basically threw those MHC, those libraries at T cells, and then they used flow sorting to detect which T cells would react and bind to the PMHC that they had. Then they found those. There's an example of if it's high on the PMHC axis and high on the CD8 axis, that means you have the PMHC is reacting and binding to T cells. And they took all the T cells and they sequenced them. It's really cool that we can do that. So they did TCR sequencing for things that reacted to the reactive peptides. And then we can see things, I don't know if you can see it, but it says CDR3 alpha, CDR3 beta. Those are 2 of the 6 loops of the TCR that interact. They're the 2 that interact most with peptide. And right there, there's a sequence of the TCR protein that reacts to SARS-CoV-2. Maybe, I don't know if you know someone who's had it. I actually have relatives who've had it. And they possibly have this TCR sequence somewhere in them. So this is the computer prediction binding algorithm that they used. And I did remember correctly, there are about 96 different peptides that the computer said, I think these might bind MHC. It just scanned them looking for patterns. And we possibly will use this later on. Here are the top 10. Here are the bottom 10. The thing is, they were able to try all of these to see what made good PMHC single chain trimer complexes. And you can see that they're kind of all over the place. You just try everything. The ones that were at the top of the list did tend to have high yields. Those are shown by the dark gray squares. But even one at the bottom of the list, at number 91 right there, that had a high yield. You don't really know, and that's why you try different stuff. So the yield column, by the way, is the protein gel band intensity. They literally ran it on a gel, and they saw how much they got. Again, stuff you've done in biochem. This is what Mary did. And so this is where I got the idea to throw this in, because she posted this on Instagram. And right here, these are the actual cells. Now, these are 293 T cells. These are making a virus that will infect T cells. So they're using these to make a virus in the lab that they're going to infect T cells with. And those T cells would make TCRs. And they were testing to make sure their system worked to make TCRs that would react with a particular PMHC. This is a validation step. It's, not the most prominent step in the whole thing, but it's an important bedrock foundation to making sure that this works. And these cells started it off. I also would like to point out, viruses can be used for good. So they had the whole stretch of the spike protein, and these are the 96 boxes are the 96 different peptides they tested. You can see that you have a bunch of different peptides. That the ones that are lit up are the ones that worked the best. And they come from all throughout the spike protein. If you look above my head, the red parts are where the activating peptides came from. And so they tried those, and they ran each PMHC is shown on a gel. You can see the yields right there are pretty high. How much was expressed, that's a reflection. You see the higher, the darker the gel, the higher the band on right below it. And then right below that is the computer predicted peptide MHC affinity. Lower is better. And again, the computer seems to do a pretty good job. It's like 50-50 of whether it will actually be good for like the top 20 that you have here. And what they found out eventually is several of these peptides actually activated T cells from donors. The top 2 are T cells from 2 different donors. There's 2 bars because they did it at 2 different times for the disease progression. The important thing is here you see at least 5 or 6 T cells, and they're shared among the 2 patients that react to the same 5 peptides. You look down at the bottom, you can see the peptides from the SARS-CoV-2 spike protein that we have caught the T cells from infected patients reacting against these. Good news, the immune system works. It does what it's supposed to. And in fact, we can see exactly where the atoms are. Once you take biochemistry, you can draw out what that peptide looks like. You know where all the atoms are. You don't know how it moves or how it binds necessarily, but you do know its shape. And that's where we start with all of this. So the conclusion is, I want you to become more and more familiar with the TCR peptide MHC architecture. Okay, 3 molecules that come together and different chains and all that. Realize that because we know the structures of these, structural immunology means we can build these in the lab. We can make them recombinantly in bacteria, and then we can actually even use those in tests or therapies. And using these recombinant proteins, we've found several spike protein peptides that activate T cell responses, and we know 5 or 6 sequences that the T cells go after. This will allow us to understand the course of the infection, understand why some people get sicker than others, and possibly help us design a vaccine. Because if we want to train T cells to go against these areas, let's give people these peptides. That's the basis of it. Thanks for sticking with me. As usual, I went longer than I needed to, but that is